Today we're doing a deep dive into, well, it's a bit of an ecological horror story. You've got one of Australia's ancient apex predators, the freshwater crocodile, and it's being brought to the very brink by, of all things, a common toad.
It really is an astonishing premise. The croc, this ultimate survivor, just completely taken out by an amphibian.
It sounds like something out of a movie.
Exactly. But the cane toad, or Rhinella marina, is arguably one of the most destructive invasive species on the entire planet. It's not just another food source, it's more like an ecological enemy, kind of biological booby trap for Australia's native carnivores.
And they have no defense against it.
Zero evolutionary defense. They've never encountered anything like it.
And the really tragic part, the irony, is that we did this. We authored this whole disaster.
We did. The toad was brought to Queensland way back in 1935. The idea was it was imported from South America by way of Hawaii with this goal of controlling cane beetles.
In the sugarcane fields, right. A noble goal, but
totally misplaced. That project failed, right away. The toads didn't touch the beetles, but they absolutely loved Australia. It was paradise for them. So they've just been colonizing relentlessly, spreading through Queensland, the Northern Territory, and now they're pushing west, hard.
An unchecked invasion.
And it has caused these catastrophic, just system-wide declines in native predator populations.
So that's our mission today. We really need to unpack this whole disaster. We're going to get into the sheer chemical power of the cane toad's toxin, look at why the traditional control methods just completely failed.
Against a population in the hundreds of millions.
Right. And then we'll get to the cutting-edge science, stuff like behavioral conditioning and even genetic engineering that might be finally turning the tide.
And for you listening, what we really want to do is break down why this invader is so lethal. We'll explain how its own body chemistry is now being turned against it, and how scientists are, using learning theory to teach these incredibly complex predators a life-saving lesson right out in the wild.
Okay, so let's start with the weapon itself, the core of this destruction. Lethal toxic ingestion, or LTI. Why are Australian animals so incredibly susceptible? Why does a crocodile die when an animal from, say, South America might just get a stomachache?
It's all about that evolutionary mismatch. Australian native species, they have absolutely no co-evolutionary history with the bufonids. That's the family of true toads. So when a predator sees this new thing, this toad, it just tries to eat it like it would any other frog.
But it's not any other frog.
Not at all. It's laced with these chemical defenses. They're called bufadinolides, bufotoxins.
So what does that poison actually do when it gets into the bloodstream of a croc or a quoll? What's happening on a molecular level?
This is what's so fascinating and so brutal. The poison works on the heart in a very precise way. These toxins, they specifically attack the engine of the heart muscle cell, which is called the sodium-potassium pump.
Okay, the sodium-potassium pump. Is that the thing that manages, like, the electrical charge in the cell?
Exactly that. Think of it as this little energy-driven pump that keeps everything in balance. The bufotoxins essentially just jam it. They gum up the works, and its activity plummets.
And then what?
Then you get a disastrous cascade. Once that pump slows, the cell floods with sodium. That then messes up the sodium-calcium exchange, which leads to this sudden, massive calcium overload inside the heart muscle cell.
A calcium overload?
Right. And that overload is what directly causes a decrease in cardiac contractility. The heart can't pump properly. It's not a slow decline. The end result is rapid acute heart failure.
Wow. Given how fast and just total that mechanism is, the impact numbers must be staggering. Are we seeing any natural resistance top up?
The initial impact is just, it's catastrophic. Because the learning curve is one shot. You try to eat the toad once and you die. So the data, the heart data, it shows this massive destruction, especially among the large frog-eating predators. For the freshwater crocodile, Crocodilus johnstoni, we've seen populations plummet by more than 70% in some river systems.
70%? And I'm guessing it's not just the crocs taking this hit.
Oh, no. It's the same pattern across the board. The northern quoll, which is this beautiful carnivorous marsupial, they've suffered declines sometimes over 75, even 90%. It's led to local wipeouts in places like Kakadu.
And the big lizards? The goannas?
Same story. Monitor lizards like Varanus panoptes have seen declines from 71 up to a staggering 97% in some river systems right after the toads moved in.
97%. When you pull the top predators out of an ecosystem that fast, that violently, it must just send the whole system into chaos.
A trophic cascade, right?
Precisely. That's what the scientists call it. Ecological turmoil.
Mm.
All of a sudden, the animals that were being eaten by the quolls and the goannas, their populations just surge.
Well, there have been observed spikes in brush turkey populations, for example. Skinks, too. It's an indirect consequence of losing the lizards that used to keep their numbers in check. And this isn't a short-term blip. The research shows these ecosystems can stay destabilized for a decade or even more after that first invasion wave passes through.
So this is a long-term
A
wound.
very long-term wound. And if you were hoping this was a problem that was, contained, well, the toads are still spreading. They are incredibly efficient invaders.
And they're getting faster, too. It's like this unstoppable, self-improving force just marching across the continent. Where's the big front line right now?
The big focus is squarely on Western Australia. The latest models, they paint a pretty grim picture. They're predicting that the toads are on track to colonize up to 75% of the Pilbara region.
The Pilbara, that's a huge, vast area.
It is. And they're predicted to do it within the next 30 years. Given how fast that front is moving, that timeline is deeply, deeply concerning.
And the Pilbara has this incredibly unique biodiversity. What do they think is going to happen when the toads finally get there?
It's predicted to be another major ecological catastrophe. The research suggests 25 native species will probably show population declines. And, critically, 9 native mammals and reptiles are likely to be added to the threatened species list.
Oh, wow.
And the ghost bat, which is already vulnerable, is projected to be pushed into an even higher threat category.
And there's a human side to this too, isn't there? The Pilbara isn't empty land.
Absolutely. The animals most at risk, like the big goannas, the blue-tongued skinks, they are so culturally important to the traditional custodians there. They're sources of food, material, they're central to storytelling and bush tucker practices.
And there are economic impacts too.
For sure. The mining sector in the Pilbara could face, significant new costs and conservation requirements because the threat status of all this local wildlife is about to change.
And here's the part that makes it even worse. The toads themselves are evolving in real time. They are actively getting better at invading.
That's right. Natural selection is working at incredible speed. It's favoring the toads that are extreme dispersers. So the populations right at the invasion front, they have different traits.
Like what? How are they different?
They move further with each hop, they travel in straighter lines, and they're way more nomadic than the older, more settled populations back in Queensland.
So the invasion front is like a self-optimizing biological machine.
That's a perfect way to put it.
Okay, before we get to how we fight back, I want to touch on a little glimmer of hope we found in the sources. A rare biological loophole. Not everything is defenseless, is it?
No, and it's a vital point. There's this small group of native Australian rodents, the water rat, the dusky rat. They seem to be physiologically resistant to the toxins.
Really? So they can just eat them?
They can. Lab studies show they can kill and eat the juvenile toads without any problem. They just see them as another food source. And because they don't get sick, they don't develop any aversion, they just keep hunting them.
A natural check and balance.
It is. A surprising one. Though, it's probably not on a large enough scale to stop the whole invasion by itself. But it's something.
Okay, so let's talk control. For decades, the main effort was, well, it seemed pretty inefficient. People doing toad musters, hand collecting them, building little fences.
Yeah, and against a population that's maybe close to 2 billion, those methods were incredibly labor-intensive. And, they just couldn't deliver long-term relief on a landscape scale.
So the thinking had to shift.
It did. Scientists started looking for high-leverage control points, weak spots. And they found one in the toad's aquatic life stage, which has led to this surprisingly successful technique using, of all things, cannibalistic traps.
Cannibalistic traps. Okay, you have to explain that.
It's this amazing biological twist. Older cane toad tadpoles are deeply cannibalistic. They actively hunt down and eat freshly laid eggs from their own species.
How do they find them?
Based on chemical cues in the water. And here's the brilliant part. The cue, the chemical they're smelling, is the toad's own toxin, the buccodylides.
No way. So the poison itself is the dinner bell.
It's the dinner bell. It signals where the fresh eggs are.
That is just incredible chemical ecology. So how do you use that to trap them? Is it scalable?
It's highly effective in targeted areas. Researchers are baiting these special funnel traps with pure adult toad toxin, the exudate from their glands. and the cannibalistic tadpoles just swarm in.
And the results?
They're phenomenal. Field trials have captured tens of thousands of toad tadpoles this way, with almost zero bycatch. The native species aren't attracted to it, so it's a great way to reduce the local toad numbers, especially in water bodies that are really important for vulnerable native animals.
So that's clever field science. What about the really high-tech, long-term stuff? Genetic tools.
Yeah, this is where a lot of the investment is going now. There's sort of two main avenues. First is genetic control of the toads themselves. There's an active research project trying to refine gene knockouts to, for instance, create non-metamorphosing tadpoles.
Meaning they just never turn into toads.
Exactly. They never leave the water, so they can't spread. Or another idea is to create toxin-reduced adult toads, which would make them harmless to predators.
And the second path is, well, it's more controversial, but it could be a lifesaver for the native species, genetic rescue.
Exactly. Instead of changing the toad, you change the predator. Scientists are looking at using tools like CRISPR and what are called induced pluripotent stem cells, iPSCs, to engineer toxin resistance genes directly into northern quolls.
So you'd be giving them the evolutionary advantage they never had.
That's the long-term, very ambitious goal, to eventually, ethically release genetically resistant quolls back into the wild.
So those genetic solutions are probably years away from being used. But there is one thing that's working right now, a deployable game changer that's acting as a lifeline for apex predators as we speak. Conditioned taste aversion, CTA.
Yes. CTA is basically taking behavioral learning theory and applying it directly to conservation. It's a natural adaptive mechanism. You teach an animal to associate a new food item, the taste of it, the smell of it, with getting immediately and severely sick.
A traumatic but non-lethal experience.
That's the goal. You want them to learn this thing will make you horribly ill. Never ever touch it again.
And this was actually trialed on freshwater crocodiles in northwestern Australia. I mean, logistically, how on earth do you teach a lesson to thousands of wild crocodiles?
It was a massive effort, led by Dr. Georgia Ward-Fear and Professor Rick Shine. They deployed almost 2,500 baits across 4 river gorge systems. And the bait was the key. They took toad carcasses, physically removed the lethal natural toxin, and then they injected them with lithium chloride.
Lithium chloride, what does that do?
It causes extreme nausea and vomiting, but it isn't fatal. So they effectively created a non-lethal poison bait to teach the lesson.
And did it work? Did the crocodiles actually learn fast enough to save themselves?
They learned incredibly fast. The monitoring showed that the number of toad baits being eaten fell, significantly and rapidly over just the 5-day trial. On day 1, almost all the baits were eaten. By day 5, in the best spots, almost none of them were being touched.
But they'd still eat other things.
Yes, and that's crucial. They kept eating the control baits, like chicken meat, just as much as before. So the aversion was specific only to the toad.
But the real test, the real measure of success, is whether it saved their lives when the real toxic toads showed up.
And it did. The difference was just stark. In the untreated control areas, crocodile mortality from eating wild toads was extremely high after the invasion wave came through. But in the areas where they'd done the baiting, crocodile mortality from toads was non-existent.
Wow.
The intervention worked. It buffered the crocodile populations right when they were at their most vulnerable.
So the strategic lesson here, especially for the teams facing that incoming invasion in the Pilbara, is about timing.
It is the most valuable insight from the whole project. They found that repeating the baiting in later years led to an even faster aversion response. So the crucial lesson is that you have to apply this CTA training right before or at the exact moment the toad invasion front arrives.
You stop that first catastrophic die-off before it can even begin.
You get ahead of the wave.
So our journey today has taken us from a catastrophic man-made biological booby trap to a really sophisticated scientific counterattack using everything from chemical ecology and tadpole cannibalism to molecular genetics and behavioral psychology.
And while the cane toad remains this massive, seemingly unstoppable ecological challenge, especially as it moves into the Pilbara, the long-term data really suggests that recovery for the native species is possible. It's going to be a race, though. A race between these human-led interventions, like CTA, and the slow grind of natural evolution.
Which leaves us with a final provocative thought for you to think about. If an animal's survival depends on either evolving resistance or learning aversion to this deadly new invader, and given the incredible speed of the toad invasion, which approach do you think will prove to be the faster, more effective path to ecological recovery in Australia? Natural selection or a well-timed human-induced lesson?
Something to think about. The strategic power of a timely lesson.
We'll talk to you next time.